In this screencast, we'll continue our review of basic quality control procedures for genome-wide association studies, focusing now on identifying and excluding poorly performing samples. We covered QC steps to identify poorly performing markers in the previous screencast. For many of the procedures we discuss in this section, you'll need genome-wide data. Since we have not provided you with genome-wide data, we'll simply review the code and discuss the results, rather than having you run the analyses on chromosome 12 only. First, we'll exclude samples with low genotyping rates using the MIND option. A high missing rate is indicative of poor DNA sample quality. So here I've gone back to the virtual machine and I've typed in the set of commands that will read in the file cleaned for Affymetrix and write out a file called cleaned5 Affymetrix, excluding any individuals with a missing rate greater than 5%. So this is this MIND option, and then the parameter you pass is the threshold of missingness. You'll exclude any individuals with a missing rate higher than the parameter you pass. So we'll run this. And as you can see in our case, nobody was excluded for a high missing rate. The next quality control step is to check for concordance between self-reported sex and genetically inferred sex. This can help identify potential sample swaps when the phenotype information does not match the genotypes. Of course, to infer a sample's sex from genotype data, one needs genotypes from the sex chromosomes, which we have not given you. So we'll walk through the commands and the results here. Here is the plink command to perform a sex check. You have to specify the binary input files as well as a name for the output file. This command will generate a file called outputfilename.sexcheck, which has 6 columns. So pedsex is the self-reported sex of the sample as recorded in the pedigree file. SNPsex is that inferred from the chromosome X data. This inference is based on an F statistic, which is a measure of homozygosity. The F statistic is a measure of homozygosity for the X chromosome SNPs. Since males have only one copy of the X chromosome, the number of heterozygous SNPs along the X chromosome is near 0, so F should be close to 1. Females have 2 copies, and on average the observed number of heterozygote SNPs should be equal to that expected under Hardy-Weinberg equilibrium. So the F statistic should be close to 0 for most females. F is of course measured with some sampling error, so Plink will flag samples with F greater than 0.8 as male and F smaller than 0.2 as female. So going back to our data example, we see 2 samples that are flagged as problematic. For one, in the 4th row, the self-reported sex is male, but the F value is quite low. This suggests that either there has been a sample swap, that in fact the DNA that was genotyped for this subject did not come from that subject, or that the DNA sample has been contaminated. The second sample flagged as problematic lies in more of a gray zone. The pedigree file lists this sample as female, but the F-test lies just outside the usual range. If you look at a lot of samples, this will eventually happen just by chance. It is not necessarily indicative of a sample swap or poor DNA. Whether to include or exclude this sample is up to the investigator. For the record, none of the samples in our example data sets fail the sex check. The next quality control analysis calculates estimates of identity by descent between pairs of samples using genome-wide SNP data. This is useful to confirm expected duplicate samples, identify unexpected duplicate samples, and identify close relatives. 2 alleles are shared identical by descent if they can be traced back to a recent common ancestor. In this case, these 2 siblings, these 2 brothers, share one allele IBD at the depicted locus because they both inherited an A allele from their father. So the fraction of their alleles at this locus shared IBD is 50%. The genome command in Plink will estimate several quantities. Z0, Z1, and Z2 are the probabilities that sample A and sample B share 0, 1, or 2 alleles at a random locus. pi is the fraction of alleles shared identical by descent. We can use Z0, Z1, and Z2 to draw inferences about sample duplicates, sample contamination, or relatedness among samples. Here are the theoretical values of Z0, Z1, and Z2 for different types of sample pairs. Here's the Plink code to calculate IBD. Note that the min option here asks Plink to only output those pairs of subjects that have pi hat over 5%. This is extremely useful, otherwise Plink would write out a row for every pair of samples, which is typically a very large file indeed. For quality control purposes, we are only interested in pairs with a pi hat greater than 10%-ish anyway. One useful summary of the IBD information is to plot Z1 versus Z0. This allows us to visually identify sample duplicates as well as particular relative pairs. So when we compare these IBD estimates with their self-reported relationship information, we confirmed the first degree relationships. So in the AFI data, 5 parent-offspring pairs were identified and 1 sibling pair, and there were 2 sibling pairs in the OmniExpress data. These genetically inferred relationships are concordant with what we knew from the PED file. Notice that if there were sample duplicates, or identical twins, they would fall in the lower left-hand corner of this plot, where Z2 is equal to 0, where Z2 is equal to 2, and Z1 and Z0 are equal to 0. Some of our downstream analyses are sensitive to the presence of pairs of close relatives, most notably the principal component analyses for population structure. So we're going to exclude one sample in each of these first degree related pairs using the remove option. So we've already created 2 files called samples exclusion OmniExpress and samples exclusion Affymetrix that contain one individual from each of these pairs who we'll exclude. So this is what the samples exclusion Affymetrix file looks like. It's just a list of the 6 individuals that we're going to exclude. And we're going to exclude them using the remove command. So remove, and then you just pass the file that lists the individuals you want to remove. And there we go, 6 individuals were removed. Finally, we'll plot the sample heterozygosity rate, or the fraction of SNPs that are heterozygous genome-wide. This is also very good at identifying contaminated samples, as they'll show up as outliers. So here are the results. Here we've plotted the inbreeding coefficient F on the y-axis for each sample, and we've ranked the samples from lowest to highest. The most important thing to note here is that because F depends on Hardy-Weinberg expectations for heterozygote proportions, it's population specific. So we've calculated F separately for the European and African samples. The second thing to note is that there's no hard and fast rule for declaring a sample an outlier. In the Europeans, there do not appear to be any outliers, but in the Africans, there may be a tail of samples with extreme heterozygosity values. Whether that's due to population heterogeneity in the African sample or to sample contamination, we don't know. We have not excluded any of these samples from downstream analyses, but you might want to flag these 1 or 2 samples at the extreme tail for further scrutiny.